Hi, Michelle Glass here. Welcome back to another one of our Chapter 22 Part 2 lectures. Our topic for this video is how we get our CD8 T cells activated. So we're looking at our immune response, which is going to be initiated by a self cell, because remember, CD8 T cells are going to recognize and bind to class 1 MHC protein. Okay, so to begin, we're going to start out with an infected body cell. Now you might want to try drawing the drawings, or you might want to just write out the steps depending on what you want to have in your notes. So you don't have to necessarily draw out these drawings. All right, so here we're looking at one of our body cells, so a somatic cell. So we know that it is going to have the MHC class 1 protein markers hanging off. And here I'm showing an organism, Streptococcus pyogenes, infecting this cell. And so we're going to then get to our first step in initiating our immune response, which is, of course, antigen presentation. Now what's happening here is our infected self cell is going to be making new MHC protein markers, and when it does that, it's going to incorporate some antigen from that Streptococcus pyogenes. It's going to hold it out like a flag, making sure that we get our immune response initiated. Now I've drawn this like a little puzzle piece because remember, we're going to have specificity to our immune response. So we're going to see those CD8 T cells recognizing specifically this antigen marker for Streptococcus pyogenes. Our second step that happens is the recognition and binding. So because we are holding out our antigen using class 1 MHC protein markers, it is going to be our CD8 T cell, which recognizes and binds. Again, you have millions of different types of CD8 T cells in the body, able to recognize and bind to the millions of different possible antigens that you're going to be exposed to. So this is our second step in our immune response. The third step that happens is what's called co-stimulation. And in this case, you can consider this like the safety on the gun. So this is where that infected self cell gives the go-ahead to that CD8 T cell. Yes, I am infected. You have my permission to kill me for the greater good of the body, right? So here is our third step in our immune response. And then as soon as you've had co-stimulation occur, then those CD8 T cells are going to activate. An activation of a lymphocyte means it's going to divide. In this case, we're going to see that the activated CD8 T cell divides to make a cytotoxic T cell and a memory T cell. That cytotoxic T cell is the one that's going to be active on this first exposure. It's our quick response. It is going to be, remember, our cell-to-cell mediated defense is going to go around identifying other body cells infected with Streptococcus pyogenes and kill them. The memory T cells are not doing anything on this first exposure, but they are going to be present so that the next time you see that antigen, you are ready to have a quicker response than you do that first time. Our cytotoxic T cells are going to be doing a variety of attacks. So we will see that cytotoxic T cells can release perforins. We saw these being released by NK cells. Remember, they're poking holes in that infected cell, and this is going to be an effective way to kill that cell. We also see that cytotoxic T cells can release Lympho Toxin. So this is named because it's coming from a lymphocyte and it's toxic to those cells. So that's going to trigger cell death. And then another way that we can target those infected self cells is those cytotoxic T cells can release chemicals in the class called cytokines. And these cytokines can trigger what's called apoptosis or cell suicide. All right, we have kind of our whole picture here, right? So remember, we have your self cell that's infected, and then step 1, we're going to do antigen presentation, followed by CD8 T cells recognizing and binding. Then you have co-stimulation as your third step. The fourth step is that activation of that CD8 T cell, which means it divides to make our cytotoxic T cell. You have actually another type of CD8 T cell that's a little bit slower about recognizing and binding. This one is going to divide to make a regulatory or suppressor T cells. They're kind of slower in their response, so they're allowing that initial immune response to occur, and then they're coming along kind of on the tail end, releasing again another group of those cytokine chemicals. In this case, they're acting as suppression factors. So they're going to help to moderate your responses of your cytotoxic T cells and also B cells, which we'll be talking about in the next video. So this is a way to have a control, because clearly these cytotoxic T cells are able to, roam the body and attack any of those cells that have been presenting this antigen for Streptococcus pyogenes, but we don't want it to get out of hand. We don't want these cells to start attacking our normal cells. Okay, that's it for now.